Hello, everyone. This is a video showing you how you're going to do your semester inquiry project, which is a project where you'll be collecting data all year or all semester, and you'll actually set up a real experiment and you'll be forming conclusions from what you test, and you get to pick it. So here's some information about it that I wanted to share. You will be working on this the whole semester. You'll have different due dates as you go along. You'll be using this presentation, this template. When I linked it, I did link it as a view only, and that is so that you make a copy of it so you're not all on the same copy. So each of you will have your own copy. You just have to make it and then turn it into the box. You'll need to turn in different slides each time there's a due date. You can turn in the whole presentation, or you can just do those slides that you needed to complete. I would rather you do the whole presentation because then I can look back and make sure everything is aligning. So the whole presentation would be best. So you do need to do an experiment on something involving a living thing, since this is biology. Some examples would be plants, seeds, yeast, worms, fish, or other invertebrates. And you have to be able to collect quantitative data, meaning you need to have numbers of some sort. So you can do mass of something, the height, the number, or the circumference, or some other parameter, but you have to be able to collect numbers. You also have to have a treatment and a control to compare together, or have two variables that you compare together. And you also need to collect the data yourself. So here is the first thing that you'll do. These dates are wrong because they're an old file, but the dates on your template are correct. Okay. So here are 5 examples of testable questions that I came up with. All of these are questions that I could set up an experiment and test. So that's what I call a testable question. What you need to not have on here is Googleable questions. Those would be questions like, why are plants green? That is not worded in a way that you could set up an experiment. You could alter it and turn it into a testable question, but these are all examples of testable questions that you could actually do. You'll do that the first week. I'll look over them, and then you'll pick one of them to design an experiment for, and that will be the following week's assignment. So let's say I picked the first one. Then you're going to do the next 3 slides for the next due date. The purpose is a 1-sentence statement explaining the main idea of an experiment, and it includes both independent and dependent variables, which you'll have on a later slide. So this purpose that I wrote for this is to determine the effect of fertilizer on plants' growth. Notice that I put the purpose of this experiment is to determine. That's how you're going to start all your sentences, like all your purpose. I mean, your purpose starts with that phrase. And then you will do your hypotheses. There are 2 kinds of hypotheses that we use in science. One of them is called the null. That's the one we're actually testing. That states that the treatment will not have an effect. And then alternative is the opposite. So for this, the null would be fertilizer will not have an effect. And then the alternative will be fertilizer will have an effect. So you'll write those 2 hypotheses, and then you'll go to your variables. So you have 4 different types of variables. Here they are. The first one is independent variable, and that is your treatment or your cause or the experimental group. So in this case, it'll be the fertilizer, the group of plants that get fertilizer. Your dependent variable is plant growth, and you can measure that in height in centimeters. So that is a quantitative measurement. And then your control is the no fertilizer. And then the constants are all the things you keep the same, like pots, soil, water, access to water, or access to sunlight, type of seed, size of seed, color, pot, etc. Everything you can think of that you need to keep the same so that you're isolating that one variable of fertilizer. So you'll do those 3 slides, and then on the next due date, after I've looked over your design, then you will move on to the procedures. So these are written step by step as if you were following a lab that you might have done in high school or college. And you need to be really specific about what you put into the, what you write up. It should be so good that somebody else could pick up these procedures and do the exact same experiment and get the same results. Oops, so there are your procedures, and it doesn't have to be a certain number of steps, just put as many steps as you need. Then after a period of time, you'll set up your experiment and then eventually you'll get some data. So here's an example of the data table. Let's say I had 10 plants set up and these are the measurements of them, and these had fertilizer, and then the right column did not have fertilizer. And then you take an average because it won't make sense to graph all of this. It's just too messy. There's too much data. So the next step is your photos. And did I put any? No, I thought I put little plants. You will put actual photos here, but this is an example. I've got 10 plants in each group, and that's because I want to be really sure of my results. If I only grow one plant in each group, then I may not be certain that the fertilizer is having an effect. It could be just that one plant is like a really, really big plant and the other one is a dinky plant. So having lots of replicates is really important. Okay, then you will do a graph. You can use Google Sheets to set up your graph. And I only graphed the averages. So if you look at the last row on my data table, I only put the averages on there. I've got my y-axis labeled with plant height. I've got my x-axis labeled with fertilizer and no fertilizer, and then I have a good title. You need to have a graph like this. If you need help, let me know. You can also watch YouTube videos of how to make a graph, but this type of graph is pretty simple. Then you will later write your results. The results either reject or fail to reject the null. And in this section, you talk about numbers and explain the graph in words. So here's my example. I have some numbers here. I could have rounded that, but I said the fertilized plants grew an average of that. Control group was that over the 2 weeks. And then I also said the data rejected my null because there was a difference. So remember, my null said there is no difference between the treatment and control, but I found that there was a difference. So I would say that I rejected my null. You cannot say that you accept your null. That's like against the rules. That's why it says fail to reject the null. Those are your two options. You can reject or you can fail to reject. Then finally, you'll write your conclusion. This is where you talk about your overall takeaways from the study, why it matters, what are the flaws, what should be done differently next time, and how can we apply this to everyday life. So here's an example of that you can read through. When you are all done at the end of the semester, you will record a video kind of like this one and present the whole project to me. And that's what your final grade will be for the lab. If you have any questions, please let me know at any point. I'll be helping you along the way. And I hope you have a great time doing an actual inquiry experiment, which every human on the planet should do at least once. So here's your chance.